Okay, so now we're going to be talking about this term osmosis and what it means, how it works. So the way I like to describe it is actually to start with this image that you should already be familiar with if you just looked at the last video clip. And if you remember, here we identified this as the simple diffusion of carbon dioxide. And remind yourself of these vocabulary words. Simple diffusion is a type of passive, so we're moving from high to low. No energy is required, and no protein help is needed. Okay, so simple diffusion of carbon dioxide. What happens with osmosis, how it's different, is that instead of carbon dioxide, we're talking about H2O, which is water. So we're talking about water moving from high to low without protein help. Okay, so let's go ahead and work through a couple of these exercises in your notes to understand what's going on here. Okay, so let's say we have a solution, and let's say it's 95% sodium. Well, then how much percentage water is it? 5. Okay, let's look at this. 10% sodium. Well, how much water? 90. Okay, it's pretty simple so far. So we've answered this first part. What is the percentage of water on each side of the membrane? Now this is where osmosis comes in, because normally, if we were talking about passive transport, normally sodium would move from high to low if we're talking about transport. But, and that means that the membrane has to be permeable to sodium. That means it has to have a transport protein to let it across. But let's look at this case here. If the membrane is not permeable to sodium, so there's an imbalance, well, what's going to happen? Well, this is when water moves. And water will move from where water is high to where water is low. So water in this case would move from the right side of the membrane to the left side of the membrane. Okay. So again, this is the principle. If there's an imbalance, but something other than water can move, it will. Water stays put. But if the thing that is in imbalance, in this case sodium, cannot pass this membrane, there's no protein to help it across, then water will move passively without protein help. So let's look at this problem here. Now let me help you interpret this image. So these dark blue dots are water, okay, and then the big pink dots are some sort of solute, sodium, glucose, could be anything. Okay. Now this is the information that's important. It's not permeable to the solute, so this membrane means these big pink dots can't get across, but it is permeable to water. So now as you're looking at this, what's going to happen? And if you think you know, go ahead and pause right now, write it out. We'll talk about it in a minute. If you're not sure how to think about this problem, well, the big thing to happen is you have to identify where things are high and where they're low. So you would ask yourself, is there a higher amount of water on A than on B? So identify where water's high, water's low. Okay. So it turns out, and you can tell this by the spacing, okay, these water molecules, they're packed tight together. There's more on A than they are on B. So if the solute can't move, water moves from its high to its low, and this is actually what's going to happen. The water will actually move from A over to B, and the volume of the water will actually decrease. And so what you notice now is that the water molecules and the solute molecules are equally spaced on either side. And so that means we've reached what's called equilibrium. It's equal on both sides. Okay, so the big question is, why does osmosis matter? Why do we care about this? Okay, and we care about it because of these things right here. If a solution has, well actually I should show you this picture right here. So what's going on here that you should notice is that this, if it's a cell, cells are packed with solutes. They're packed with molecules. And a vast number of these molecules cannot get across this plasma membrane. And so that means that water is going to be moving either in or out of the cell depending on what's outside of the cell. Okay. So, if we have a situation here, and this is an image from your book, if we have a situation here where there's the same number of solutes and water inside the cell as outside the cell, then it's called an isotonic solution, and water overall doesn't move. And this is showing you a happy red blood cell. This is really important for your blood. But here we have a different situation, and I think I actually have a better image of it here. Well, no, these are the same images, but it shows this writing here, which I think is helpful. So here the salt or the solute concentration outside the cell in that solution is less than the salt inside the cell. This is called an hypotonic solution, and what's happening is that water, in an attempt to balance it out, rushes into the cell. And that causes the cell to blow up and eventually burst open because so much water has rushed into it. Now, here's a different situation. There's more salt, there's more solutes outside the cell than inside the cell. So what that means is going to happen is all the water inside the cell is going to rush out of the cell, and the cell crinkles up. And that's called crenation. Okay, so this is the way I remember it, and you're going to have to see my face to see this. So I think hypo, my lips go hypo. And so I think that's what's happening to the cell. It's water's rushing in, and the cell goes, oh, and it blows up. Okay. Here's the opposite, hyper. I think hyper, and so that's what the cell does. It crinkles up. And so I think of the shape of my lips to think what's happening to the cell. Now, just to remind you, so hypotonic causes the cell to lyse, which means blow up, okay? And hypertonic causes the cell to what's called crenate, which means it crinkles up due to water loss. Okay? So you need to know these vocabulary words. Now, why does that matter? What happens in each of these situations? Well, isotonic red blood cells, normally they're going to be carrying oxygen around our blood, and we need these cells shaped a certain way because they are going to fit single file through many of our smallest blood vessels called capillaries. Well, if they blow apart, they can't carry anything. And so we lose our oxygen carrying capacity of our blood. Here, if they crinkle up, you can just imagine these things, they don't flow right, they start hitting our blood vessel walls, and these little spiky things can start tearing things up. So these don't carry oxygen as well either, and they can really start to cause problems to your blood vessels. Okay? And so what that means is if you're giving an IV as a healthcare professional, you better get the right type of solution. You better get an isotonic solution that has just the right amount of salt in it. Hypotonic would be pure water. If you gave that to your patients as an IV, you're going to kill them. Okay? And so knowing these things and what they do to cells is going to be very, very important as you think about some of your duties as a healthcare professional.